Okay, everyone, welcome to the first video lecture for Chapter 6 on cell structure and function. This chapter is fairly long, so there's going to be at least 2 videos for this, possibly 3, okay? All right, so here in Chapter 6, we're talking about some really important stuff in biology, the structure of cells. So what do you find inside of cells? How's it all put together? How does it relate? How do these various parts of the cell that we're going to talk about, how do they work together to allow for a functioning living cell? All right, so a couple of things about cells. First of all, they are the fundamental units of life. Okay, this means that they are essentially the smallest, or simplest rather, collection of matter that can be alive. If you take the cell by itself, cells do all the things that living things do. They reproduce, they interact with their environment, they can sense their environment, they process energy, all these sorts of things. They tick off all the boxes that we see as requirements for something to be alive. Now, when you look at all the different components inside the cell that we're going to talk about throughout this chapter, none of them, in and of themselves, are considered to be alive. Okay, they don't do all the things that living things do. But when you put them together in a watery environment, they begin to interact with each other, and you wrap all that in a cell membrane, you get a living thing. So it's very difficult to sort of describe what life is. So what we generally do is we define something as living or not based on what it does. And cells, of course, do all these things that living things do. But again, the things inside the cell don't necessarily do that. But when you take all these parts and put them together in the right way, life emerges. So we talked about emergent properties in a previous chapter. You take simple things, build them into more complex assemblage, and at some point, as things become more complex, new properties emerge. Well, life is an emergent property, and it shows up first at the level of the cell. Okay, so a couple of things more about cells. The way that we talk about cells, the way that we understand cells is sort of encompassed in what we call the cell theory. Okay, cell theory. Cell theory basically says that, it says 2 things really. It says first that all cells are related by descent from earlier cells. So all the cells in your body came from earlier pre-existing cells. Cells don't just spontaneously appear out of nowhere, out of nothingness. They come from pre-existing cells. Okay, so that's point number 1. Second point is that the cell is the fundamental unit of life, as we talked about, right? So these 2 things then come together to comprise what we call cell theory. Now, by the way, just because it says theory doesn't mean there's any question in our minds about whether or not cells exist. We obviously know that they exist, and so don't take that word theory to mean something other than it actually does. All right. Now, as we're going to see throughout this chapter, even though cells are quite different from each other in terms of external appearance, they're basically the same internally. They all have the same basic structural features, okay, for the most part. So as an example of what I'm talking about with regard to looking different in terms of external appearance, take a look at these 2 cells. What you see here, this sort of orange or lighter orange colored cell, that's a neuron, a nerve cell, okay? These that I'm highlighting now, these long sort of red structures, those are muscle cells. So these 2 cells are very different in terms of external appearance, but internally they're much the same, actually, okay? But their outward physical differences result in them doing dramatically different things. Muscle cells are used for movement, motility, for contraction, movement of body parts, limbs, things like that. The nerve cell is there to transmit nerve impulses from one part of the body to the next. In fact, you can see how the nerve cell has contacted the muscle cell here, for example, at that junction, and that's going to allow the nerve cell to pass an impulse into the muscle cell, which will then cause the muscle cell to relax or contract, whatever the case might be. Okay, so basically we have two main types of cells on the planet. All cells can be lumped into two very broad categories, okay? First of all, what we have are eukaryotic cells. So eukaryotic cells are animal cells, plant cells, cells of fungi, protists, single-celled organisms you would find in fresh and marine waters, for example. Those are the kinds of cells that our bodies are made of, okay? The second type of cell is the prokaryotic cell. Prokaryotic cells are bacterial. They are bacteria. All bacterial cells are prokaryotic. So we have this fundamental distinction then between these two main categories of cells. All cells on the planet currently can be lumped into either the prokaryotic or the eukaryotic category. Now, in terms of prokaryotes, okay, we have really two main categories. Prokaryotes or bacteria have been around for a very, very long time. There is fossil evidence that bacteria existed as far back in Earth's history as 3.8 billion years ago. That's billion with a B. Nothing like a eukaryotic cell shows up in the fossil record until around maybe 2.5 or 2.6 billion years ago. So bacteria had the planet to themselves for a very long time, and so they had a much longer time to evolve and diversify. Now, when you look at most bacteria today under a microscope, they all kind of look the same. There's not a lot of external physical difference between them. But when you look at them genetically, when you compare their DNA sequences, we see two major categories of prokaryotic cells, or two major domains. So domains are taxonomic categories, okay? And they're part of our classification system. The domain is a higher level than the kingdom. So you may remember from high school biology, kingdom, phylum, class, order, family, genus, species, the categories that we use in taxonomy to classify organisms. Domain is a relatively new category. It sits on top of the kingdom, so it's larger than that. And when you look at bacterial taxonomy or classification, we see that there are two domains. There's domain archaea and domain bacteria, okay? Now, this is just a way to distinguish the genetic differences between these two major categories of bacterial or prokaryotic cells, all right? Now, in terms of eukaryotes, so eukaryotes, I should have written this before, belong to domain eukarya. So we have three different domains: eukarya, archaea, and bacteria get two of them. So this should tell you that there's much more diversity, genetic diversity, within the prokaryotic groups of organisms than there are within the eukaryotic groups of organisms, okay? In fact, life on Earth is probably best described as bacterial, even though when you look around you today, you see humans and animals and plants and things like that, the big things. There are far more bacteria around than there is anything else. And they've been here longer, and they will be here well after everything else is gone. So you can sort of think of eukaryotic life on this planet as sort of this little offshoot off of the prokaryotic line. And that kind of, we're not used to thinking of things that way. You know, it kind of makes humans less significant, but in the grand scheme of things, it's fundamentally true. Okay. So let's now start talking about the structure of cells, okay? So when we compare eukaryotes and prokaryotes, we see that they do share some things in common, right? So what do they have in common? Well, they always have a plasma membrane. A plasma membrane is sort of the outer boundary of the cell. We talked about it a little bit in chapter 5. We talked about phospholipids, and I said phospholipids come together to make a cell membrane, or a phospholipid bilayer. That's what a plasma membrane is, okay? It's a phospholipid bilayer. You find it in prokaryotes. You also find it in eukaryotes. Inside the cell, there's sort of a semi-fluid substance called the cytosol, It's also referred to as cytoplasm. That's just the term that I learned. That's the term that I generally use, cytoplasm. But cytosol, cytoplasm, whichever term you use is fine. It's the watery environment of the cell. Then we have chromosomes. So chromosomes are chunks of DNA. They contain genetic information, all right? Now, typically bacteria have one circular chromosome, one circular DNA molecule. Eukaryotes have their DNA chopped up into many more pieces. For example, humans have 46 chromosomes or 23 pairs. Don't worry about that right now. Just think of a chromosome as a chunk of DNA that contains genetic information. Prokaryotes and eukaryotes both have those. And then finally, the last thing really that's common between prokaryotes and eukaryotes are ribosomes. Ribosomes are protein factories, all right? They do one thing and one thing only. They make protein. And you find them in abundance in both prokaryotic and eukaryotic cells. So these four things then are common to both cell types, both eukaryotes and prokaryotes. And that's really where the similarities stop for the most part, okay? All right, so let's start talking about those differences then, all right? Now, we're going to see, and in fact, we're going to spend much of the rest of this chapter talking about eukaryotes because they're so much more complex than prokaryotes, and frankly, they're the kinds of cells that we're made of, so there's some interest there on our part. We're going to see there's lots of different things inside the eukaryotic cell. By and large, prokaryotes don't have those things. All right, so what you're not going to see in a prokaryote is a nucleus. There's no nucleus. In eukaryotes, as we'll see, the nucleus contains the DNA. It separates the DNA of a eukaryotic cell from all the rest of the internal environment of that cell for very good reasons. Prokaryotes don't have a nucleus. Their DNA is basically just located in a region of the cell called the nucleoid. Okay, nucleoid. The nucleoid is nothing more than a region of the cytoplasm where you find the DNA sort of congregated or collected or concentrated in a prokaryote. There's no membrane around it separating it from anything else. Excuse me. We also, in eukaryotes, have structures called organelles. Okay, you don't find those in prokaryotes. Organelles are organ-like structures. In the same way that in your body you have, kidneys, a spleen, pancreas, liver, stomach, and so on, all these organs that do very specific jobs, very specific things, we have in eukaryotes organelles, organ-like structures that carry out very specific functions. You don't find those in prokaryotes, all right? Okay, in other words, the take-home message from that is that prokaryotes are much simpler than eukaryotes. So let's take a look at a typical prokaryote here. Let's look at the drawing on the left here first. So this is a typical rod-shaped bacterium, all right? We're going to start from the outside of the cell and kind of work our way inward. First of all, what you see here is these little hair-like structures. These are called fimbriae. Now, not all bacteria have these, but a good many of them do. And fimbriae are structures that allow bacteria to sort of attach to surfaces or attach to each other and collect together in colonies and that sort of thing, all right? Now, there are other structures similar to that, but unless you're taking a microbiology course, it's not really important here. First layer, outermost layer that you see here in yellow, okay, that is called the capsule. There you see the term, it's capsule. This is a fairly thick layer. It's made up of carbohydrates primarily, so it's sort of like a sticky layer. It also helps bacteria to sort of collect on surfaces or attach to surfaces and that kind of thing. Inside of that, this is where it gets a little bit hard to see. The layer just inside of that is the cell wall. Cell wall is sort of that middle layer you can kind of see right there. All bacteria, all prokaryotes have a cell wall. The cell wall is sort of a protective layer. Think of it as sort of like a suit of armor. It helps to protect the cell from mechanical damage, from being torn open and that sort of thing. Also helps to maintain the cell shape, okay? You find cell walls in some eukaryotes, plants in particular, but you don't find them in animal cells, other types of eukaryotes, okay? But all prokaryotes have a cell wall. Inside that, we have the plasma membrane, or the cell membrane. It's the innermost layer here, okay? The cell membrane is really sort of considered to be the boundary between the living internal environment of the cell and the non-living external environment. Now, inside of that, you have the fluid volume here, okay? The fluid, again, is called the cytoplasm or cytosol. And inside of that, or floating in that, I should say, you have these little dots. These are ribosomes. Now, there are a lot more ribosomes in a bacterial cell than what this figure is sort of showing. There are hundreds of thousands, if not millions of them inside a typical bacterium. And what they're doing is producing protein. So it's not, too far off the mark to think of bacteria really as nothing more than protein factories. That's what their major job is. They produce proteins, and they're exceedingly good at it, okay? Now, finally, this large purple area, that's the nucleoid. So that's the one chromosome that bacteria typically have. If you took that out and sort of laid it out flat, you would see it's a loop. It's a large loop of DNA, and it gets kind of mashed up into a ball to fit inside that cell. And the region where you find that, again, is called the nucleoid, all right? Okay, last thing, these extensions off the back end of this bacterium, these are called flagella. Typical bacteria will have, one or several of these, depending. Not all of them have them, but most of them do. And these are outboard motors. If we kind of zoom in a little bit, you can sort of see that this flagellum goes through the cell wall, or the capsule rather, into the cell wall and into the cell membrane, and anchors there. There are a series of proteins there that act like a motor. They go through a chemical reaction, they interact with each other, and it causes the base of that flagellum to rotate or to spin. And so what that does is it causes the flagellum to rotate, and as it gets further away from the cell, the arc of rotation gets wider and wider, and so it's whipping around like an outboard motor behind the cell, and it allows that cell to swim through water. It powers it through the water of the environment it's living in. All right. Now, here we have an electron microscope view of this. You can't see everything we talked about, but most of the things you can. For example, you can see the very, very thick capsule layer here on the outside. That sort of white line you're seeing there inside of it, it's probably the cell wall. The membrane on the inside of that you probably cannot see. You can see a dark layer here. That's the nucleoid where the DNA is. This is the cytoplasm, this fluid-filled space. And then you see this sort of grainy appearance. It's grainy because of all those ribosomes that I talked about. That gives you a good idea of how many ribosomes are there. You can't see the flagella in that image, but they are there. Okay, now let's shift over again and talk about eukaryotes. And we're going to spend the rest of this chapter talking about eukaryotes. So a couple of things about eukaryotes that really distinguishes them from prokaryotes. First of all, they do have a nucleus, and the nucleus holds the DNA. It contains the DNA of the cell, all right? They have membrane-bound organelles, membrane-bound organelles that act like organs of the cell, basically performing very specific functions. These are really the two key things that distinguish eukaryotes from prokaryotes, okay? Eukaryotes also have cytoplasm, but again, that's something common between them and prokaryotes. Another unique thing about eukaryotes is that they're generally much larger than prokaryotes. By volume, on average, maybe 100 to 1,000 times larger than a typical prokaryotic cell. And they're also a lot more complex in terms of what's going on inside of them, what kind of structures they have, the functions that they carry out, all that sort of thing. Okay, so now let's talk about the plasma membrane a little bit. Okay, now this again is something that's common between prokaryotes and eukaryotes. Think of the plasma membrane, let me just draw a line here. The plasma membrane is a selective barrier. What this means is that it is a barrier between the external environment and the internal environment, but it's selective in the sense that it allows some things to move across fairly easily, but not others. So it's what we call selectively permeable. So this term permeable means, if I tell you something is permeable, that means that things can move across it. But a cell membrane being selectively permeable does not allow everything to move across it. So it's a filter, basically, all right? And in that way, by controlling what gets into the cell and what gets out, it keeps the inside the cell chemically very different from the external environment. What you have inside the cell is the chemistry that you need for life. It's a very special kind of chemistry. You generally don't have that outside the cell. So you have to have a selectively permeable membrane, the cell membrane, to maintain those differences, to maintain a distinct chemical environment inside the cell versus outside, okay? And the way that cell membranes are made is essentially by a phospholipid bilayer. Remember, the phospholipid, and we talked about this in the last chapter, is basically a fat molecule that has a polar head that attracts to water and nonpolar fatty acid chains, two of them, that do not attract to water. So the polar head is hydrophilic, the nonpolar tails are hydrophobic, and so when you put these in water, they automatically sort of do this, right? Remember we talked about that? They back up to each other in this way and form a bilayer structure that you see right there, with water inside the cell, water outside the cell, the heads attract to water, the tails fold inward and face inward, back up to each other to exclude water. So you end up with this fairly thick hydrophobic region in the middle of the cell, and that hydrophobic region is largely what makes this membrane selective. Anything inside or outside the cell dissolved in water has a difficult time getting through that fat layer, because you know that oil and water don't mix. They form layers. They separate from each other. They repel each other. So that fairly thick hydrophobic region in the middle, where the tails are of the phospholipids, is a lot of what gives that membrane its selective permeability. So let's take a look. First of all, what you're seeing here is an electron microscope view and cross-section of a cell membrane. So this is the inside of the cell, that's cytoplasm. Here's the outside of the cell, and what you see there, it almost looks like a cross-section of an Oreo cookie. That is the phospholipid bilayer. The white layer you're seeing in the middle, in other words, the cream of the cookie, right? That's the hydrophobic tails. The two dark layers would be the heads of the phospholipids that are pointing inward toward water and outward toward water. Now, the figure below shows you the phospholipid bilayer here, okay? So you've got water, let's say, outside the cell, water inside the cell. Here's your hydrophobic interior of that cell membrane with the heads facing water on both sides. Also, what you see here are various different kinds of proteins. These proteins, in some cases, as you see in this example, act as channels. They allow things to move back and forth through that membrane. So if something is dissolved in water inside the cell, if it's dissolved in water, it's by definition polar, right? Which means it's going to have a difficult time diffusing through that non-polar hydrophobic layer. Just like water and oil don't mix, anything dissolved in water won't mix with oil or fat. And that's what you have here. So this protein, called a channel protein, allows for that solute to move through without encountering that hydrophobic region that would prevent it from moving through. So that's one of the very important functions of membrane proteins, is to act as channels, allow things to move through that wouldn't be able to get through the membrane otherwise. And there are lots of other functions of these proteins that we'll see as we go forward here. Okay, next thing we're going to talk about is the underlying reason why it is that cells are by and large microscopic. The vast majority of cells on the planet are microscopic, okay? Now, that's not true of all of them. It's certainly true of bacteria. It's true of most eukaryotes, but not all eukaryotes. The largest cell on the planet is actually an ostrich egg. It's the size of a bowling ball, basically, but it is a single cell. That's true of all other eggs as well. So chicken eggs, any other kind of egg you're talking about, it is a single cell. So clearly some of them are large enough that you don't need a microscope to see them, but they tend to be the exception to the rule. The vast majority of cells are microscopic, and whenever you see sort of a commonality like that in biology, there's generally a good reason for it, right? It's not just random. There's a very good reason for that. And there is a very good reason why most cells are microscopic. It has to do with the logistics of moving things into and out of the cell, okay? So we have a really important concept in biology in particular that you'll see throughout all levels of biology, from single-celled organisms all the way up to ecosystems, for example. And that concept is surface area, scratch it out, surface area to volume ratio. So when you're talking about a cell, there are two things that you can measure about it. Excuse me. You can measure the amount of surface area of the membrane, how much membrane there is, and you can measure the volume inside the cell. So when you talk about the surface area, you're talking about the cell membrane. Think of the cell membrane as the gateway. If something is going to move into that cell or out of it, it has to go across that cell membrane. All right? I'll grab a drink here real quick. All right, sorry guys. So think of the membrane, the surface area of the membrane, as the gateway, the doorway. If something's going to go into or out of the cell, it has to go through that membrane. Now, that's important. Keep that in mind. If you compare, let's say, a small cell, so I'm just going to draw a cube here to sort of represent a cell, a small cell, and you measure the amount of surface area there and measure the volume, you would see there's a certain amount of surface area relative to volume. And again, that is the surface area to volume ratio. Now, if you compared the surface area to volume ratio in the small cell to a large cell, okay, you could do the same calculation here, and we'll do this in a minute. You would actually see that this ratio, the ratio of surface area to volume in the small cell, is larger than it is in the large cell. All right. Now, the key to understanding this is that surface area, being two-dimensional, grows by the square. Volume, being three-dimensional, grows by the cube. So do the math. Take the number 2 and square it. 2 times 2 equals 4, right? So 2 times 2 equals 4, right? 2 squared equals 4. Take the number 2 and cube it. 2 times 2 times 2 times 2, what's that equal? 8, right? That's an 8. So which of these mathematical operations, squaring or cubing, gives you the larger number faster? Cubing, because you have 3 dimensions growing at once, not just 2. Not just length, width, and height. Okay? So the point I'm getting at is, let's say that as this small cell grows and becomes a larger cell, the surface area and the volume do not grow at the same rate. The surface area grows by the square, but volume grows by the cube. So which of these two is growing faster? The volume. That means volume, or the pace of volume growth, is outstripping the pace of surface area growth. That means that this cell, relatively speaking, has a larger amount of surface area compared to its internal volume than does this cell. Because as the cell grew, this surface area grew more slowly than the volume does. And so if you're thinking of that surface area, again, as the doorway, the gateway to that cell, which you should, this larger cell has a smaller doorway and a larger volume, whereas the smaller cell has a larger doorway and a smaller volume. And so things having to get across that cell membrane, this smaller cell membrane relative to volume can become a bottleneck, a choke point. And if you think of a cell as sort of like a factory, which they essentially are, each one having on average maybe 20 or 25,000 chemical reactions going on per second, they need a lot of materials coming in, raw materials coming in to do what they do. And they produce a lot of products that need to be shipped off to the outside. They're like a factory. So here you have a factory with a really large doorway for things to come in, large enough to get all that stuff in to power what's going on in that volume. Here you have a large factory with a small doorway. You can't get enough stuff in fast enough to supply all the operations going on inside. And so that large cell may not survive. this is why cells are by and large microscopic. Now, what about these large cells like eggs I talked about? They have very specialized adaptations that allow them to get around these kinds of problems. In fact, when you compare eukaryotes to prokaryotes, one of the reasons why eukaryotes have membrane-bound organelles, prokaryotes don't, is that those membrane-bound organelles are an adaptation to increase the surface area of all the membrane while not increasing the volume so much. It helps to get around this problem. So let's take a look at this. This is a figure from your textbook. So I'm just going to show you the math here to prove to you that what I've just said is true. So here we have cubes, right? So imagine this is a small cell. And just to make it simple, let's say it's 1 millimeter on a side. It's a cube, right? Okay, how would you determine the total surface area of that cell? Well, first of all, you'd find the surface area of one side. What's the surface area of a square? Length times width. So 1 times 1 gives you 1 square millimeter on a side. Cube has 6 sides, so 6 times 1 gives you a total surface area of 6 square millimeters. That's what that number says, right? 6 square millimeters. Now, volume. Volume is length times width times height. So 1 times 1 times 1, 1 cubic millimeter. That is 1 cubic millimeter. So what is our surface area to volume ratio? Divide your surface area by your volume. 6 divided by 1, 6. So our surface area to volume ratio is 6. This means that for every 1 unit of volume, there are 6 units of surface area. That's a big doorway. Plenty of surface area relative to volume. Now, surface area increases. Let's imagine we now have a cell that's 5 millimeters on a side. Surface area of one side, 5 times 5, 25 square millimeters, times 6 sides, 150 square millimeters. All right, volume, 5 times 5 is 25, times 5 is 125. Okay, you see this is much larger volume than we had in the first cell, but look at our surface area. Whereas this is 125 times that, this 150 is not 125 times that. It's 25 times it, right? So take your surface area of the large cell, divide by the volume of the large cell, 1.2. 1.2 means there's 1.2 units of volume now. I'm sorry, 1.2 units of surface area for every 1 unit of volume. Over here we had 6 units of surface area for every 1 unit of volume. And that's because our surface area grows faster than our volume does, okay? So this large cell has much less or much smaller doorway or gateway across which things can move to supply the much larger internal volume. So it's a bottleneck. It's a choke point, essentially, in the flow of materials into and out of the cell. Well, how do eukaryotes get around that? They increase their surface area without increasing their volume, and that's what membrane-bound organelles are all about, increasing the amount of surface area of plasma membrane without increasing the volume. So take that same large cell and now divide it up into a whole bunch of 1 cubic millimeter volumes. Now you have dramatically increased the amount of surface area but not change the volume. Surface area divided by volume, back up to 6. So now we're good, right? That's a fundamental distinction between eukaryotes and prokaryotes. That's why eukaryotes have, one of the main reasons why I should say eukaryotes have a lot of internal membrane-bound organelles. It's an adaptation to get around this surface area to volume logistical problem, okay? All right, surface area to volume ratio is a really important concept at the level of the cell, but also at all other levels of complexity going on all the way up to ecosystems. It plays a fundamental role in the way that things are structured, the way that things are built, designed, so to speak, by evolution. Okay. All right. So now we're going to get into talking about what it is we find inside of eukaryotic cells. Okay. So plant and animal cells have most of the same organelles. Mostly they're the same, right? They are eukaryotes, so that shouldn't be too much of a surprise. They do very different things, though, and largely the differences that we see between plants and animals have to do with the fact that plants carry out photosynthesis and animals don't, and the fact that plants don't have any sort of animal-like skeletal system like what you find in animals. So those two things are the real reasons why animal and plant cells differ. So let's take a look. This is a typical animal cell. I'm just going to kind of go through this in a very broad overview because later on in this chapter we're going to hit all these different components. Okay, so let's sort of work our way from inside to out. What you see here is the nucleus of an animal cell. It contains the DNA. Chromatin is a term or a name for DNA. Okay, we'll explain why we use a different name for DNA, chromatin, a little bit later on, but the nucleus is what contains the chromatin, a form of DNA. Okay, and you see that sort of as these threads of material here. There's also a structure inside the nucleus called the nucleolus. The nucleolus makes a special kind of RNA, ribosomal RNA, that is used to build ribosomes, the protein factories. All right? And all of this is housed within a nuclear envelope, a membrane. All right. Outside of that, we have this large sort of jelly roll looking structure shown in blue here. All right. This is called endoplasmic reticulum. In particular, it's rough ER, rough endoplasmic reticulum. It's called rough because it has ribosomes in it. If you look, you can see these little dots in that membrane. Ribosomes do one thing, like I said, they make protein. So the rough ER is a protein factory. And so you see how it's looped and folded like that. Whenever you see looping and folding of a membrane in a cell, or anywhere really, that is an adaptation to increase the surface area of that membrane. It tells you that membrane is important. The more that membrane the cell has, in this case, the more proteins it can make. So having a lot of surface area of membrane there is really important. We have a second type of ER, endoplasmic reticulum, called smooth. And you see it right here. Now, it is not looped and folded like that. It's sort of a meshwork of tubules that run in all three directions, almost like cargo netting, I guess you could say. It's another way to increase surface area, but notice it's smooth. There are no ribosomes on it, so it doesn't make protein. It has various other functions to play that we'll talk about a little bit later on in the chapter. Okay. Outside the smooth ER, or the rough ER rather, we have another highly looped and folded organelle called the Golgi apparatus. Think of the Golgi apparatus as sort of like the FedEx of the cell. It's a shipping and receiving center. Essentially what it's going to do is take protein products that are made by the rough ER, take them in, sort them, modify them, and ship them off, typically to the outside of the cell. So think of it as sort of a distribution center. Okay, we'll see more about that a little bit later on. All right, here we have organelles called mitochondria. Oops, sorry. Organelles called mitochondria. There's one, there's another. Usually there are dozens to hundreds of these inside cells, depending on cell type. But think of the mitochondria as sort of like the generators of the cell. They're going to produce energy in the form, or a form, that the cell can use. Okay, we have an entire chapter, chapter 9, on cell respiration, which is the process that these mitochondria use to make energy make energy from our food in a form that the cell can use. Notice that inside there's a membrane that again is looped and folded, right? That tells you that membrane is very, very important. The mitochondria needs as much of that in there as it can possibly get. Okay, here we have a structure called a centrosome. There you see the term right there, centrosome. This is an organelle that is sort of like an organization unit inside the cell. Basically, it produces fibers. You see these fibers kind of growing out of it? They're called microtubules, okay? The centrosomes really come into their own during cell division. Those microtubule fibers will grow out from that centrosome and they'll attach to chromosomes and push and pull and line them up and pull them apart and distribute them to the two halves of the cell that will eventually become two separate cells. So they're very important in terms of making sure that once cell division is complete, the two cells that result have a complete set of DNA, all right? I tend to think of them as like traffic cops of the cell. They're regulating movement of chromosomes, in particular during cell division, all right? Now, along with those microtubules that come out of the centrosomes, we have two other types of filaments, microfilaments and intermediate filaments. All three of these fiber types are protein. They're made of protein, different kinds of proteins. Collectively, we call them the cytoskeleton. Cytoskeleton means cellular skeleton. So they make up this sort of three-dimensional protein network that runs throughout the inside of the cell. It's not really shown very well in this figure, but it has a number of different jobs. First of all, like I said, microtubules help with cell division. The microfilaments and intermediate filaments help to anchor things in place, move things around during various aspects of the cell's life, run around the inside of the membrane, shoring up the membrane, making it stronger, all kinds of things, and we'll get to them toward the back half of this chapter. All right. One thing that the microfilaments do in particular is that they push parts of the cell membrane outward into these projections called microvilli. Not all cells have these, not all animal cells, but a good number do. And these microvilli increase the surface area of the membrane for absorption. We'll talk about why you need that a little bit later on. Basically, the more membrane of a cell that is coming into contact with food, let's say in the lumen or the space of the digestive tract, the more membrane in contact with food, the better the absorption, right? And this is a way to increase surface area without increasing volume, because if you increase volume, you run into that problem, surface area to volume ratio we just talked about. All right, peroxisome that you see here. A peroxisome is an important organelle. It helps to basically degrade hydrogen peroxide. Peroxide is a byproduct of some of the cellular metabolic processes, and it can be very dangerous to the cell. Peroxisomes will help to get rid of that, but they can also do various other things as well. Okay, a lysosome, this guy right here, a lysosome. Lysosomes are actually produced from the Golgi apparatus. They contain proteins that act as enzymes, hydrolytic enzymes. Remember, hydrolysis means a reaction that breaks things down, right? So hydrolytic enzymes break things down. Lysosomes can recycle worn-out organelles. They can digest particles that might find their way inside the cell and break them down. They're sort of like the garbage crews or refuse crews of the cell. Okay, then let's see. Over here we have a flagellum, right? So we talked about flagella on bacteria. Well, this is a eukaryote. There's one kind of eukaryotic cell that has a flagellum that it uses to swim with. It's sperm. It's animal sperm. Also, some plant sperm have flagella as well, but typically you find them only on sperm cells. So these are the basic parts of an animal cell. Again, we're going to cover each of these in more detail throughout the rest of the chapter. Let's go on next and talk about what we find in plant cells. So plant cells, again, have basically the main things that animal cells do. Let's go over those again real quick. We have a nucleus with a nucleolus inside. We have chromatin inside, DNA. We have a rough ER, smooth ER, ribosomes, all that. Golgi apparatus as our shipping and receiving center, the FedEx of the cell, if you will. Mitochondria, generators of the cell, peroxisomes, all that. All right, we have a cell membrane, again, just as an animal cell does. Okay, a phospholipid bilayer that separates the internal environment, the living internal environment from the external environment. So all those things are pretty much common between plant and animal cells. We have cytoskeleton, again, okay, it does the same basic things as it does in an animal cell. Now, what's different? Well, chloroplasts. Chloroplasts are the large green structures that you see right there. Chloroplasts are responsible for running photosynthesis. They're why plants appear green. We don't have chloroplasts, right? You can't go stand out in the sun as an animal and get your lunch that way. You have to consume your lunch, right? Okay, we'll talk about chloroplasts later on. Another thing that distinguishes plant cells from animal cells is this large structure here. Think of it as a bubble of membrane inside the cell. It's called a central vacuole. Basically, it's a big water tank. It stores water, dissolved minerals, nutrients, things like that. You find these in plant cells. The only kind of animal cell you see them in are in mammal adipose cells that store fat. Okay, they have basically a central vacuole that stores up those fat droplets. But other than that, you don't see central vacuoles in animal cells. Think of them as a plant thing. Then we have this large layer here, this thick layer here outside the cell membrane. That's the cell wall. You never ever have a cell wall in animal cells. Okay, they have a cell membrane, but no cell wall. They're two fundamentally different things. A cell wall is a plant thing. You do find them in fungi, but it's very different differently made in fungi compared to plant cells. All right, the plant cell wall rather, is why plant cells have this very angular shape. Usually are shaped like bricks, rectangular, cubic, that kind of thing. Animal cells take on a wide variety of different shapes because they don't have cell walls. Think about the muscle cell and the nerve cell I showed you at the beginning of this chapter. So the plant cell wall is very rigid, it's very tough, it's like a suit of armor. It essentially does for the plant cell what the cell wall of a bacterial cell does. But again, those cell walls are very, very differently constructed. There's no evolutionary link between them. They're very different, okay? All right, finally, we have these connecting channels, almost like holes have been drilled between adjacent cell walls that combine, or rather link, the inside of one cell to the next. They're communicating channels. They're called plasmodesmata. They allow for things to move back and forth, molecules move back and forth, allow for adjacent cells to communicate, coordinate their activities, and that kind of thing. All right? Okay. I've been going for about 45 minutes, so we're going to stop here, and I'll pick up with, we'll dive into more detail to these various parts of the cells in the next video lecture, the second part for this chapter.